Okay, well, I think we've got quite a good audience here. So it's my pleasure to introduce Dmitry Matyushov. Dmitry graduated from the Moscow Institute of Physics and Technology in 1986, and then did a PhD at Kyiv State University, which I'm using the Ukrainian pronunciation, not the Russian, Kyiv State University and the Ukrainian Academy of Sciences. And then from a series of research positions and distinguished postdoctoral fellowships, came to ASU in 2000. My knowledge of Dmitry came from a discussion with the famous physical chemist Mark Ratner, who said to me, you know, you've got the world's leading expert on electron transferring proteins at ASU. No, who's that? So you never know what your colleagues are doing. And I remembered that many years later, just a couple of years ago, when my lab started getting involved in electronic measurements on proteins, and we were staggered, as a few other people who've done these kinds of things have been, by the remarkable electronic properties of proteins, and the fact they make incredible designer electronic components, which are things you might not expect. And Dmitry indeed has proved himself to be this world's leading expert, because he has now produced, in collaboration with others, an all-atom quantitative theory of this process, which actually appears to work. And if you had told me, actually at any time in my lifetime, that doing quantitative calculations of this sort on something as complicated as a protein was possible, I would have thought we were mad. So maybe Dmitry's mad, but he's very clever mad. Dmitry.
Thank you, Stuart. Well, it's clearly a great pleasure to present at home. It's probably my second seminar here at Physics. Unfortunately, I lost my voice in our cold climate. And so if I need to speak up, please let me know. Hopefully, I will manage. So again, as local, I understand that this is a colloquium, so I need to talk to essentially to physics undergraduates. So that's what I will try to do. I hope I will still be able to entertain some more sophisticated of you. So I will be talking about some statistical rules that we believe should apply to biological systems, and in particular to biological function. And in the end, I will talk about some very recent studies, particularly from Stuart's lab, which most of them are still not published, but they are leading to some, I believe, very exciting results. So I need to define my subject. And again, I assume that people here do not know much about biology. And so we are talking about biological function. What I mean by that is really a molecule known as an enzyme, which is usually just a large protein. And there is a very little spot inside of this protein, which is actually responsible for the entire action, and it's called the active site. So what happens in the active site is a miracle, and a chemical reaction is being catalyzed, which means it goes faster than it's supposed to be. And that's what we call the catalytic effect, really. So what it means in like textbook description is that you have the barrier separating reactants and product, and enzyme somehow manages to lower that barrier. The speed up factor is absolutely amazing. It's about 17 orders of magnitude for fastest enzymes. Why does it happen? We still mostly do not know. The widespread kind of qualitative picture was introduced back in 48 by Linus Pauling, still in most of the textbooks around, which tells you that protein somehow creates a field which favors that state on the top of the barrier. So once you go to that position, there is a field created by the protein which makes the barrier lower. And so there are some typical numbers that you find in the textbook. So the lowering of this activation barrier results in this enormous speed up factor. These are not the only enzymes that you will see in nature. There is a very significant set of enzymes which are responsible for the energy produced by biology. And so all energy that biology is using is produced in so-called energy chains. It's either respiratory chains in animals like in us, it would be in mitochondria, or in photosynthesis, They are pretty similar, and the ideas behind those chains are probably concepts are very similar as well. So you have some very large protein complexes sitting in the membrane. Those are, as a matter of fact, the largest protein complexes that we know of. And they essentially help to move electrons in one direction across the membrane, and protons are actually in the opposite direction, and the protons in the opposite direction. So they will create the gradient of protons, which eventually goes through the ATP synthase and drives this famous reaction, which essentially stores all the energy. The same happens in photosynthesis. So essentially, in this case, you get the energy not from food, but you get the energy from a photon. For plants, you have two photosystems, so it means two photons come in, and you have this famous Z scheme of photosynthesis. But the idea is the same. You move electrons across the membrane. You create a difference in concentrations of protons, and that eventually is used to store the energy in the chemical bond. So I will be talking about ideas behind efficiency in biology, and I will look at efficiency in a very narrow sense. It will be essentially to lower the activation barrier in catalysis. Of course, there is also energetic efficiency, which is completely separate issue. So I'm trying to narrow my scope to looking only at what we know as the catalytic effect. And I will mention briefly at the very end what can be done for the energetic efficiency. So we do not know much about how to describe this catalytic effect in terms of quantitative models, as Stuart mentioned. And the question is, maybe we can define some new statistical rules of proteins to explain the catalytic effect. And so these rules actually kind of rotate along the ideas which belong to the construct introduced by Maxwell, is known as Maxwell's demon. And they result eventually in the violation of one of fundamental relations in statistical mechanics, and that is fluctuation dissipation relations. And eventually it applies to conductivity of proteins. And I'm not going to claim that I solved all the problems, because many of them are still there. And one of the biggest ones is why do we have to have those big complexes? So this is, for instance, a BC1 complex, a famous complex in mitochondria in plants. And it's enormous, but the molecules which allow to transfer charge are those heme sitting inside. And so the question is, why is that nature decided to waste all those resources and produce such enormous molecules to move subatomic particles, electrons, and protons across the membrane? So it's still with us. We don't know why this is. All right. So because I'm going to talk about statistics, let's start with the Gibbs ensemble, and again assume this is kind of undergraduate level talk. And so Gibbs, when he was formulating his ensemble, it actually described well in his own book. He was very much concerned about how to construct the ensemble which would satisfy some macroscopic constraints. So an ensemble is essentially a collection of independent systems with the same Hamiltonian, which have different initial conditions which describe your microscopic states. But they have to be consistent with some specific macroscopic conditions, for instance, conservation of energy and so forth. And so this is how it's explained by James. So, and he realized that the problem is actually ill-posed, because in order to define such an ensemble, you actually need to know all the possible constraints, and you never know them. And so, as James put it, but Gibbs decided to be brave and ignore all the warnings and go ahead and formulate a ensemble which has only one constraint, that the temperature is constant, and neglect all the rest. And essentially, that's what came out, as we all know, the distribution of energies, which has the Boltzmann factor and partition function and the density of states. And it uses only temperature as the constraint. And the only reason we believe this ensemble is all right is that it describes all thermodynamic data. And so that's really the foundation. So my point is that in principle, you can build many ensembles. And so the Gibson ensemble is not the only solution. And that, of course, was well understood by Lebowitz, who said, well, this is just a mathematical tool. And depending on the physics that you have, you might build other ensembles. And that's what we are going to do, because we believe that there is a new physics that you need to introduce to the problem. So when you talk about constraints, you have to turn back to, again, this famous construct, which was introduced by Maxwell in his book on theory of heat in 1871. And so what this creature is doing, it essentially uses some information to create a non-equilibrium state. So the most kind of textbook example is that this demon is going to measure velocities of particles and then separate a gas into a hot component and cold component without making any external work, which means violating the second law of thermodynamics. So Maxwell himself actually noted that it doesn't have to be velocity. It can be any measurement that the demon is going to make. For instance, he suggested that the demon can measure directions of molecules if you let in only molecule flying from left to right and not allow the opposite direction. So then you create the gradient of pressure. Again, it's a creation of a non-equilibrium state without performing work. The important thing, though, is that what the demon does, it imposes some constraints on your ensemble based on the knowledge that the demon gains, right? And so, and constraints require information. So there is a close connection between information and constraints. And so people notice that enzymes, in a sense, work as Maxwell's demon, because they look at the molecules and decide, I like these molecules, I do not like the rest. So they essentially apply this kind of idea that, well, I am going to choose something and use it for the reaction. And of course, some people suggested that biology violates the second law. Well, this is a modern vitalism, of course, but its discussion itself turned out to be quite useful to kind of understand what's really going on. So the resolution of the paradox came about 100 years later, most prominently from work of Bennett, who realized that computations can be done completely reversibly without performing work. So what people were thinking about that this demon, when it measures the velocities of the molecules, essentially collecting the information, it must perform some work, and that should explain the paradox, right? But turned out that you actually can do computations or information processing without performing the work. And so the paradox is resolved by so-called Landauer's principle, which tells you that it's erasure of information that requires production of entropy and heat. So for every bit of information, the system will release natural log of two states, one bit of entropy, which means that the heat which will be delivered to the environment will be greater or equal to kBT, natural log of 2. So that kind of resolution of the paradox was actually recognized by Jack Monod, who was thinking about allosteric action of proteins. And he said, well, every enzyme, as a matter of fact, is a heat engine, which means it has to work in a cycle. And so it has to come back to its initial position where it started from. And so he actually made this, created this analogy with mythical character Sisyphus, who has to roll the rock up to the hill, but eventually the rock will roll down. When the rock rolls down, it means your enzyme is being reset. And so this repeated motion of the rock up the hill and down is really what enzyme is doing. So now we are essentially understanding that all these kind of creepy ideas can be formulated in terms of some constraints put into statistical mechanics, and we want to understand what are the consequences of constraints. And it turns out that there are many condensed matter systems which do involve constraints and corresponding statistical consequences. The most famous one is the glassy state. So when a liquid becomes a glass, it loses ergodicity, which means it cannot visit all the configurations available to a high temperature liquid. And so this system becomes non-ergodic. The way to understand it is to look at this cartoon, which represents the system as kind of landscape of the energy. So each point on this Q coordinate is some state of the system. And then, but the energy changes with the maxima and minima. And so at some point, you're getting stuck in a specific minimum because the time scale that is required to jump from one minimum to another becomes longer than the observation time. So essentially, instead of understanding the entire landscape, you're getting stuck in one village and you have no idea there is a city next to you, and all your knowledge is limited by this one minimum, which is known as a component. So at this point, your system becomes non-ergodic because it cannot explore all possible configurations. Turns out that the proteins have landscape similar to glasses, not exactly the same. People talk about different topology of protein landscapes, but the idea is pretty much the same. You have a bunch of minima, and there are some activation barriers between them. And if you heat up the protein, you actually can observe all different transitions to different scales of motions, from local motions to more and more collective motions as you allow the system to go over the barriers. So the same idea, if you get stuck in one of the barriers, in one of the wells, you become non-ergodic. So recently, probably starting from the 90s, we started to see single molecule measurements in biology. And those essentially allowed to realize that not only the single protein molecule has a complex landscape, but actually the entire ensemble of enzymes is highly heterogeneous. There are two types of heterogeneity that people are looking at. The first one is static, which means that different enzymes will have different activities. And that was realized in this beautiful experiment in which enzymes were sprayed out into oil drops. And once the enzyme does some reaction, you can see as optical signal. And only few of them showed optical signal. It means most of the enzymes sitting in those oil drops were non-active, and only few of them were active. So which means the activity of the enzyme was very different depending on which molecule you're looking at. And once people started to look at dynamic measurements with single molecule spectroscopy, you also found that there is a dynamic heterogeneity of enzymes, meaning that the rate of catalytic reaction will change significantly depending on the conformational state. For instance, this kind of cartoon shows you an open and closed state of a molecule where the rate will fluctuate significantly between different states. So the landscape is complex. The question is, well, does it matter? So we are essentially getting to the point, what are the properties of all those demons which are going to constrain the phase space or configuration space? And starting from Maxwell demon, you immediately realize that this creature, which is a macroscopic creature, should interact with microscopic degrees of freedom. So the same is for the enzyme. It's a molecule, but it needs to somehow interact with macroscopic degrees of freedom to split the phase space into components. And of course, imposition of constraints is also the property of all the demons. What does it do? It does a spontaneous symmetry breaking, like in a phase transition. Why? Because it doesn't change the Hamiltonian, it changes the distribution. So the picture here is really simple. You are sitting in some part of the phase space, let's say gamma I, and you want to jump to another part of the phase space, but it takes too long time. So because the time of the jump is much greater than the time of observation, which means that you have no chance to be there. And in order to express this mathematically, you need to put this Heaviside function in front of the Boltzmann factor. So the symmetry of the Hamiltonian is not perturbed, but the symmetry of the distribution is perturbed. And so that means that the properties of the symmetries which are already embedded in the distribution are lower than the symmetry of the Hamiltonian. We have spontaneous symmetry breaking. All right, so essentially what is happening is that I have my phase space separated in different portions or minima or components. The question is, who is that or what is that demon which is going to make the selection? And in order to understand that, it's better to look not at the reaction itself, but to resetting of the enzyme which happens after the reaction. So again, looking at Monod's picture, you have your Sisyphus, which is going to roll the rock back down the hill, and that system, which is my enzyme, is going to end up in different components. Some of them will be active, most of them will be inactive. Actually, this is an example from a already fairly old measurement, single molecule measurements on the enzymes, which shows some activity as a function of some time trace. And so it turns out that enzymes sleep 97% of the time. So only 3% of the enzymes are active and 97 are inactive. So this phase space essentially is very sparse. So there are very few points here which are active. And who is the demon? The demon is the reaction, because the reaction is going to choose only active states and leave behind the inactive states. And so essentially the reaction is the one which takes the components which will produce the product. And by using the fact that you have to be there on the reaction timescale, you actually shrink your configurational space to some small component. All right, so that all sounds good, but we of course want observable consequences. And the first observable consequence is the violation of the fluctuation dissipation theorem. Okay, I'll try to make it simple. Again, going back to the idea, this is undergraduate talk. So how do we understand the fluctuation dissipation relations of theorem? So the best way is, of course, to go back to harmonic oscillators. We always use harmonic oscillators in physics. So you pull a spring with a force, you get the extension. Of course, it's time dependent, and that will produce for you the frequency dependent fluctuation dissipation relation. But I'm interested here in the static limit. And in the static limit, I'm going to saturate, so I get some displacement delta x, which is of course related by Hooke's law to the force. So I have my susceptibility function, which is simply inverse force constant. Okay. At the same time, I can use the equipartition theorem and say, well, I can learn about this force constant not by pulling the spring, but simply looking at spontaneous fluctuations of the spring, right? So then you get the same information, but I just need to involve the temperature factor. And so the fluctuation dissipation relation essentially tells you that either by pulling the spring or by looking at spontaneous fluctuations, you're getting the same information, you're getting the force constant of the spring, okay? Is it true? Again, to bring things home, I show here a plot from Austin Angel's review article. Many glasses and proteins actually show that this is not true. So essentially what we have here is the prediction known as Nyquist theorem or Nyquist noise, telling you that MSD of some variable, and in this case, this is atomic displacement of within a bulk material or within a protein, should scale as temperature. And typically that's what you see at low temperatures. But then at some temperature close to the 200 Kelvin, which is known as the temperature of dynamical transition, you get a change in the slope. which essentially tells you that there are more fluctuations that you would expect based on standard statistical mechanics. Okay, so what do? I have more fluctuations. I need to take somehow account for the fact that the variance is greater than it should be. And so what people do about that is that they say, well, if you're shrinking your phase space of some constrained components, so in order to describe the statistics of fluctuations in that component, you need to introduce some effective temperature. So because my delta x squared is now larger than I expect, I need to divide it by temperature T effective, which is larger than my kinetic temperature, standard temperature of kinetic motion. And so that defines for us the effective temperature. And as a matter of fact, in a influential paper in Nature, Kurchan noted that that must be actually the definition of non-equilibrium state, so that you must have different temperatures if you apply different thermometers. So if you use different properties to measure the property of the temperature of the medium, they should give you different numbers, and that would be the definition of non-equilibrium state. So it's kind of a much deeper view of the problem, because it says, well, every non-equilibrium property every non-equilibrium state should have this property. This is a simple kind of example of what you do. This is a mechanical example published by Israelov. And so what he did, he used simply AFM tip placed to glassy glycerol. And so in this case, you can compare the response to the push of this tip on the substrate with spontaneous fluctuations of the tip. And then you get some effective temperature, which drops down to the kinetic temperature as you allow your glass to age. Of course, the change here is not that dramatic, so you see just slightly bigger effective temperature. So the equilibrium one will be around 180, and then here you have 184. But that demonstrates you the principle that if you separately can measure spontaneous fluctuations and response to applied force, you can figure out what the effective temperature is. Again, following the recipe that you can look at different thermometers, you can look at other properties. And of course, if you are thinking about catalysis, so then electrostatics is more important than mechanical deformation. As a matter of fact, is the most important parameter here. And then you can look at some very simple properties, and this is now done by molecular dynamic simulations. For instance, the potential, electrostatic potential at the active site of the protein. And so then you straightforward calculations. You calculate the probability density of that potential. You find the average. You find the variance. And the fluctuation dissipation relation tells you that the average should be related to the variance by inverse temperature. And what you find for proteins, again, that there is too much noise compared to what you expect. So your variance is much greater than you anticipate based on FDT. And again, you define the temperature as the ratio of the variance over the average now for the electrostatic energy. And you again find that T effective is greater than T. All right, why does it matter? You can understand why that should matter for activated transitions by looking at simple examples. For instance, I can consider a diffusion of a particle on a lattice. So you can consider a particle which is going to jump from one side to the next side, and it's going to go over some barrier. And that barrier comes from elastic deformation. So it's elastically deformed in one well, then it jumps to the other well, and this crossing point will be my barrier. How I calculate it? Very easily, I take the force constant of my spring. I need to take the displacement, which will be half of delta x, the distance between the minima. So I just square it, multiply by K, and this is my elastic deformation. That would be the barrier required to go from one state to another. But if my force constant is defined with some fluctuations which are different from statistical ensemble, from Gibbs statistical ensemble, so then again, this what happens is that the barrier that I calculated based on FDT should be multiplied by the ratio of T over T effective, and it's clearly seen here. So essentially, my activation barrier goes down because this ratio is less than 1. So what it tells me really is that if I have this effective temperature, so then immediately it follows that my barrier should, for activated transition, should go down as well. Can I use it for transferring charge within proteins? Yes, I can, because the situation here is quite similar to what happens to diffusion of particles. The reactions, typical reactions that happen in proteins is that you take electron from one side, you need to move it to another side. And that process, of course, is happening by tunneling. So it's under the barrier tunneling with the some probability decaying as exponential of the distance. But that's not the only thing that should happen. For quantum mechanical tunneling to happen, you have to have the resonance of two energies. You have to have equal energies. So otherwise, you violate the law of conservation of energy. And so there must be some fluctuations which would allow this resonance, and you need to calculate the probability of that fluctuation to happen. So which means that you essentially need to overlap two Gaussian distributions of energy levels, which are distributed because your system is connected to some thermal bath. And so I can take the Gaussian probability, take the log of the Gaussian probability, I get again the parabola. So the situation here is absolutely similar to simple elastic motion of the particle and the lattice. You have two parabolas which cross, they'll give you the barrier. And there are two parameters really which will give you completely the barrier height. And this is the distance between the minima and the curvature. And the curvature is given to you by fluctuations around the minimum. And so you can define the ratio of two parameters. They are in this business, they are called reorganization energies. It doesn't matter what the name is. The point is that there is the first parameter, which is similar to displacement. This is kind of the push on the spring and the response of the spring. And the other parameter is spontaneous fluctuations. And so the ratio of the two will give you an idea what is the effective temperature in the system. And so you can define this ratio. That's why these parameters are defined in this way. So their ratio essentially gives you immediately the ratio of T over T effective. And so what is happening is that absolutely clear from this diagram, it's very similar to diffusion and the lattice, so that your barrier goes down as the ratio of two temperatures. So the existence of effective temperature essentially allows you to have a lower barrier. So what we know is that the separation of fluctuations and response, if that happens, for a particular medium, they're going to lower the activation barrier, and that's exactly what we need for the catalytic effect. The main question, of course, from the practical standpoint, what is the ratio? Doesn't matter at all. If it's the same ratio as I showed you in that experiment with AFM tip, then it would not matter because the ratio would be very close to 1. But again, we have different thermometers. That was the mechanical thermometer. And now we have the electrostatic thermometer. And they might produce you different results, and they do. So this is an example, again, of this enormous complex 1 of biological energy chains. And if you're looking at this arm sitting in the matrix, inside of that protein, there is a chain of cofactors. So these are essentially bus stops for the electron. An electron travels from one side to another by hops. Those are iron sulfur clusters where you have a complex of iron atoms and sulfur. But the main point is that if you try to put it in the computer and again calculate this ratio of these two parameters, you find some very low number. So depending on which side you're looking something like between 0.2, 0.3, 0.4, but those are numbers which are significantly deviating from unity, which means you can expect significant lowering of the barrier, and that's exactly what is happening. Your barrier goes significantly down. So the catalytic effect of this particular enzyme can be explained by the effective temperature for the electrostatic fluctuation. All right, so what happens if you lower the temperature? On that plot that I showed you before for MSDs, we know that FDT works at low temperatures. It stops working at high temperatures. Turns out the same applies to electrostatics. Again, these two parameters, they are separated at high temperature, so they approximately a factor of 2 or 3. And if you lower the temperature, you suddenly collapse them together, which means that you are restoring FDT at low temperatures, and at physiological temperatures, you break FDT. So there is kind of consistency between those measurements and the results that we are getting for the electrostatics. But the main take-home message is that proteins, in terms of electrostatic fluctuations, operate as a glassy system with the T effective over T between 2 and 4, and it's much greater than people found for mechanical deformations. How about the bigger picture? So those processes that I told you about, those are just hops between two sites. You have two molecules and you tunnel electron between two molecules inside the protein. Can you move charges in protein over large distance? That was imagined already a long time ago by St. Georgi, who thought that entire cell acts as an electrical device. He actually thought that most of the biological function somehow is related to electrical signals, whether that's true or not, but he clearly imagined that proteins can be conductive. So essentially he said, okay, the valence and conduction bands of the protein are separated by two larger gaps so that you cannot really move electrons up to the conduction band. But what you can do is that you can essentially create holes if you pull out electrons from the valence band, so then you can create conductivity in proteins. Well, that was suggested in 1968. Stewart actually did it recently by doing exactly what was suggested. Very clever trick. So if you introduce something that can pull electrons out of the protein and place it between two electrodes, then you create holes, and those holes can migrate through the protein. So the conductivities turned out to be quite significant on the level of Simons per meter. It means if you have a nanometer object of the protein, so you have nanosimons in the conductivity. And most importantly, the conductivity was scaling non-exponentially with the distance, which means this is not tunneling, and most likely it's a sequence of hops. So there are hops of holes through the protein which are responsible for those numbers. The main question is, of course, can we account for the current? The current is something like nanoamperes. And in order to account for that current, you need to have sufficiently fast hops of the electron through the protein. The question is, can we produce a quantitative model which would account for the observable currents? That's not the only observation. The other side of the picture came from microbiologists who started to look at some bacteria living without oxygen. And they discovered conductivity through so-called organic protein nanowires. So those are actually cytochrome molecules. So cytochrome molecule is a protein which has this heme containing the iron, which can change its oxidation state. And those are placed in a continuous wires. And so what they do, they actually discard electrons. So every time you have a energy chain in biology, you have electrons which need to be discarded somewhere. So we in our mitochondria, we do it by putting electrons to oxygen. So we reduce oxygen, making water, and that's how we get rid of the electrons. These bacteria, they live in without oxygen, so they need to put extra electrons somewhere, and they send those electrons to rocks. So they connect through those nanowires to rocks and send those electrons through the nanowires to get rid of them. So that has been known already for a while. Recently, people at Yale actually measured the conductivity of those things, and that turned out to be on the same scale as Stuart was measuring for the proteins. And of course, the question is, why is that? What is the mechanism related to this conductivity? We still don't know. There are a number of groups now kind of racing to understand this mechanism. What we can say is that at least for one cytochrome, the picture is about the same as I described to you. So people actually measured single cytochromes in electrochemical measurements, in which you have cytochrome sitting on the side of next to the electrode, and then by measuring the current by changing over voltage, you can calculate essentially the conductivity. So this is the derivative of current over voltage, and it's supposed to give you a Gaussian function. So we actually try to simulate the system, and here is the MD line. So it's not a fit. Fit is actually a dash line, and MD line was simulated directly by computer, and from that simulation we again obtained the same ratio of T over T effective, about 40%. So at least there is a possibility that the conductivity of those microbial nanowires is again related to the effective temperature. We don't know yet. So most of those examples involved, and particularly in biological energy chains, they involve cofactors, essentially the stops at which electron can go to move from one side to another. The question is, do we need cofactors? Understanding of this came actually again from completely different side. As usually happens in science, different people contribute to the picture without even realizing that they do. So biochemists doing biochemistry of enzymes started to ask the question, where those enzymes put reactive holes that they create. So if there is a reaction, they can create a hole in the active site, and that hole can oxidize other components of the cell and create some damage. And so the question was, well, how do they figure out how to eliminate that damage? And so they realized that many proteins that have active site actually have chains of these two amino acids, tryptophan and tyrosine. And the current analysis shows that about 30% of known proteins have some chains where these amino acids are sitting within the distance which electron can tunnel. And so there is also a very interesting connection to evolution. So we did not have all 20 amino acids when life started. So originally there were only 9 amino acids in so-called primordial soup. So this is a famous experiment by Stanley Miller, when he created a soup and found by some extreme conditions, and found 9 amino acids in this primordial mixture. And so then more amino acids were added. At some point, we got some aromatic amino acids, and that's related to the creation of, so actually to the appearance of oxygen in the atmosphere. And the last two, tryptophan and tyrosine, I believe to be connected to the need to fight oxygen and peroxide. So the idea is that the proteins can actually conduct on their own without any specific cofactors because they have chains of those amino acids. And we are, of course, wanted to test it and try to understand whether these new statistical rules that we understand should play some role can describe what experiment shows to us. So that's a recent study again performed together with Stuart. So this protein was measured, conductivity was measured, and it was placed in the computer, and we tried to look at the hops of election between different tyrosines here. And that's the kind of the results of simulations, which show you again, you have a separation of two responses. One is spontaneous fluctuation, another is kind of response to the external perturbation, which I described in terms of these two parameters, but this is essentially the ratio of kinetic and effective temperature. And you find again the same kind of magnitude. It seems to be that all proteins are around the same magnitude and ratio between 0.2 and 0.4. And it turns out that we actually can describe the conductivity measured experimentally when we apply this effective temperature to activation barriers. So another experiment, kind of bringing us back home. All the arguments that I've done about the new type of ensemble and non-Gibson distribution functions, they clearly apply to anything that you calculate with those ensembles. And you do not necessarily have to calculate response function. You can calculate the entire Gibbs energy. And so then, of course, the change of the Gibbs energy, like Gibbs energy of the reaction. So you go from reactants to products, you have some delta G. That delta G should be also multiplied by the same factor. And we, of course, realized that, but I was a little bit scared to go that far because if you open any biochemistry textbooks, everything is calculated from here. And that non-ergodic delta G essentially doesn't exist. Until very recent measurements done again by Stuart and Bob Nemanich showed that it might be actually true. So what they did, they put the protein between two electrodes and they measured the conductivity as a function of the potential of the electrode, okay? And so that conductivity is supposed to have a peak, but as we know from kind of standard electrochemistry and thermodynamics, that peak is supposed to be sitting at the equilibrium redox potential of the pair that you have here, which for those amino acids, tryptophan and tyrosine, is about 1 volt. What they measured instead is about 0.3 volt. And again, it brings us to the same ratio, T over T effective, somewhere around 0.3. If that's true, it means that many of the calculations involving the Gibbs energy should be reconsidered. But to me, so far it looks a little bit spooky because it's way too much. But that's where we are right now. And the story, of course, is not over because there are many components which I just mentioned, but we still do not have the answer. I mentioned that Maxwell-Demon must involve information flow, and so does any working enzyme. How does it extend to functionality of the entire biological system? We still do not know. We do not know much about other thermometers, so we essentially try to look to electrostatic thermometer. Some people looked like with neutron scattering that I showed to you, mechanical thermometer, which is displacements of atoms. But you can imagine many other properties for which you can measure different properties, the mean and the fluctuation, and those should give you, presumably should give you some other temperatures. And of course, the whole question of non-ergodicity in molecular biology is still completely open. I'm glad that I'm not the only one who thinks about these issues. Paul Davis presented his book recently in this colloquium, which talks about similar issues in biology. What is the role of information? Does Maxwell's demon exist in biology and all those things? And so I'm glad that particularly on this department, I am not alone. Which brings us to the hope that we are now moving from the early stages of the field development, which can be connected to this quote from Chargraph when he was talking about molecular biology, which started with an orgy of model building. And I think we also went through this point where some of the models were quite crazy. It's also important to realize that we're actually not proposing anything new. Just look at the classics and statistical mechanics by Richard Feynman. First page defines what is equilibrium. You need to count the number of ifs in the definition of equilibrium. And the most important one, that equilibrium, I forgot to highlight another if here. And that the fast things have happened and the slow things have not. So essentially, you're supposed to sit in a window between different timescales, between fast timescale and slow timescale, and that's where you can define thermal equilibrium. The problem with most of the biology, those windows do not exist. You have so densely distributed relaxation times, wherever you go with your observation time, whether it's your neutron scattering spectrometer or measuring of conductivity in Stewart experiments, you always find some relaxation time which is sitting right top of on your measurements. And it means you're never there. You're never in equilibrium. So what I wanted to show you is that we clearly understand that biology is fundamentally non-ergodic. The entire evolution has happened as a crack in the ice, just one trajectory. There is no exploration of the phase space, right? The question is how far down it's going. So we believe that this non-ergodic property of biology actually goes all the way down to molecular biology, and of course there are some consequences for biological function and bioenergy. Jack Monod compared an enzyme to the rock, right, being pushed up by Sisyphus. And I just want to finish with the quote from Albert Camus, who said, "The rock is still rolling." And so every time enzyme does a function, it pushes the rock up the hill, but then it resets itself and it's still going on. Thank you for listening.